gentlemen, my name is Charlene Gaimoso and I will be your host for today's experiment. If you're curious what kind of experiment we will be doing this day, it is all about biomolecules and enzymes. So without further ado, let's get into knowing what biomolecules, enzymes, and what kind of experiment we will be doing in this episode. If you've ever wondered how all the food that you eat gets digested, it is not only because of the acid in your stomach that does that job. Many molecules in your body called enzymes also make possible the digestion. In fact, there are thousands of enzymes in your body that perform a variety of functions to keep you healthy and active. For example, enzymes aid in the digestion of food in your stomach. Our bodies makes enzymes which assist in the transformation of chemicals by acting as catalysts in our bodies. An enzyme catalyst starts reaction and makes them happen faster by accelerating reactions that otherwise might not occur at all or would take too long to complete. One of the common enzymes that has the ability to protect the cells from oxidative damage by converting hydrogen peroxide into a non-toxic form of liquid is what we call catalysts. The damages that may occur inside your cells is only natural due to the reactions. Me and my group formulated some questions why we want to do this experiment. Number one, what are the effects of the catalyst enzymes for each biomolecule that we provided? Number two, once you add the enzyme, does the catalyst react with the biomolecule immediately? Number three, is the result different or the same compared on each cup? Number four, does time really matter when the food is submerged into the container? And lastly, how do enzymes help in digesting food as what you have observed in the experiment? In this experiment, we provided the top 3 biomolecule that is very relevant to all of us. Number 1 is carbohydrates, second is protein, and third is fat, or this is also what we call lipid. So by this experiment, together with the enzyme, we will know it together how enzymes and other biomolecules react not only outside by this experiment and also inside our body. And of course, our group has formulated a hypothesis regarding this activity. As we know, enzymes are biochemical catalysts that helps to start and speed up chemical reactions. And the enzyme used for this experiment is the catalyst that was obtained from juicing the papaya. If enzymes really behave depending on the factors interacts with them, we can say that this experiment is one of the ways to find out because the enzyme can react into different types, sizes, and structure and a vast array of functions of biomolecule. The interaction of the enzyme in each biomolecule predictable. Some specimen may absorb the enzyme too quickly, some will deform, change in texture, color, and shape. And by doing this activity, we will prove to you if whether our hypothesis is correct or not. So what are you waiting for? Let's go and start this activity by of course first presenting to you the materials that you will be needing if you ever want to conduct this activity or experiment yourself at your homes. Let's go! Welcome back again to our experiment. So here are the materials that you will be needing if you want to do this experiment yourself. First of all, we have biomolecules. We provided here 3 biomolecules that will serve as our specimen in this experiment. The hard-boiled egg here will serve as our protein specimen. The pork chop here will serve as our fat specimen, and our bread here will serve as our carbohydrate specimen. Of course, if we have biomolecules, we will have enzymes. So we provided a pure papaya fruit juice that will serve as our reactant in this experiment. And of course, do not forget the 3 containers that we will be needing, and I also labeled them to fat, protein, and carbs so that you won't forget which container you used for a single specimen. And you will also be needing a timer. Here, for our initial experiment, our timer will be just 30 minutes. And for the last final result, you will be needing 1 hour in this experiment. So, without further ado, let's start. First things first, we will put our enzyme, which is the papaya juice, in each of the containers. So, let's do it. extra enzyme here, so we will put it on the tea bottles. And of course, after that, we will be putting our specimens on each cup. Okay, so we will be starting now. Go. Now we will start our timer. 30 minutes. It will start now. Okay. 30 minutes has already passed. So now we will be checking the reaction between our specimen and our enzymes on our initial reaction. So let's go check it. Our 30 minutes again right now. 30 minutes has already passed, and this is our final result, and we will observe what happened to our specimens after an hour. And there goes our experiment.